So let me get started. So welcome to the University of Connecticut Brain Imaging Research Center's Speaker Tag Series. I'm the host, Fumiko Haight, Director of BIRC, the Brain Imaging Research Center, and Professor of Psychological Sciences, Math, Neuroscience, and Psychiatry. This speaker series is unique in that the speaker tags the next speaker, also known, I call it, okay, I apologize, this is part 2, so there's some overlapping jokes, and I'll try to cut them, but I was going to say that I call it also a Ponzi scheme speaker series, because once you get a great speaker, you say this person spoke also, and then people seem to agree, and we've had fantastic speakers like we have today. We've had Danielle Ansari, Lucy Nodine, and many others that you see on this slide, as well as on our website. So we started this in April when the COVID pandemic happened and many of the in-person talks were shut down. And it's been quite successful with over 100 institutions joining and 1,000 people registering to our talks. And today it seems like we're having a great number of participation as well so far. And as you can see in the slide, you can view previous recordings from wonderful speakers such as Tor Wager, Kim Noble, and so on our website, as well as Danny Bassett's talk from last time. And you can also sign up to get updates on our talk series by emailing Lizzie, as you see in this email address on this slide. And I'm delighted to announce our next speaker that is coming in 2 weeks. In mid-June, who is Daphne Shahami from Columbia University, who's internationally known for her work in learning, memory, and decision-making. So as many of you are used to Zoom calls already and talks, housekeeping items are that we will keep you muted except for at the when you have questions, you can mute yourself. In general, we're taking it through the chat box, so if you can stay muted and just type in your question, that'll be fantastic. We'll generally ask at the end, especially because we need to finish by noon promptly or 5 or so minutes before, and Danny will let us know when she needs to leave. She has a meeting that she could not get out of right after this. And I'll be moderating while also admitting people, or Lizzie might be doing this as well, and checking the chat box, so I apologize if I look very distracted. So I'll get started. Today's a great pleasure to introduce for the second time Professor Danny Bassett. Danny is a J. Peter Skurkanich Professor at the University of Pennsylvania with affiliations in the departments of bioengineering, physics and astronomy, electrical and systems engineering, neurology, and psychiatry. I wonder if all of this fits on her name card, if she still has one of those, which seems almost old-fashioned these days. She's also an external professor at the Santa Fe Institute. She's most well known for her work blending neural and systems engineering to identify fundamental mechanisms of cognition disease in human brain networks. I've known her work through Mike Gazzaniga giving a talk when she was still when he was giving a keynote at CNS in San Francisco, and she was a postdoc back then with Mike Gazzaniga. I was very disappointed when he ended up giving the talk running out of time, just flashing and zooming through her slides, her work without mentioning any details of it, but I'm very fortunate that we will get to hear about that today. She received a BS in physics from Penn State University and a PhD in physics from the University of Cambridge as a Churchill Scholar and as an NIH Health Sciences Scholar. Following a postdoctoral position at UC Santa Barbara, as I mentioned, she has done amazing work at University of Pennsylvania. She has received multiple prestigious awards, including the American Psychological Association's Rising Star, Alfred Sloan Research Fellowship, MacArthur Genius Fellowship, Early Academic Achievement Award from IEEE, and so on. I'm not going to list all of them because it takes probably a couple minutes just going through all of this. She's an author of more than 280 peer-reviewed publications already, with over 21,000 citations. She's the founding director of the Penn Network Visualization Program, a combined undergraduate art internship and K-12 outreach program, bridging network science and the visual arts. Her work has been sponsored by NSF, NIH, Army Research Office and Laboratory, as well as many other organizations, and we are very fortunate to have her. Please join me in welcoming Danny. Thank you.
Thank you very much. Should I go ahead and share my screen then?
Yes, if you don't mind.
Great. You can see it okay?
Yep.
Wonderful. Okay, good. Wonderful. So thank you so much for having me yet again. And today what I wanted to do is talk about a completely different project. I titled this talk Perturbation and Control for Human Brain Network Dynamics. And I know that's a bit of a mouthful. So what I'm going to do over the next few minutes is to try to unpack precisely what I mean by each of those terms and why I think it's important to understand them in order to understand human cognition and its alteration in disease. But before I get started, what I wanted to do was to mention the fact that I feel particularly heavy today, given the events of the last few days. And I wanted to highlight the work of my colleague, Ishmael, who is a professor of biology at the University of Pennsylvania. He's a fantastic scientist. And he posted on Twitter on May 30th this quote. He says, well, my 8-year-old finally asked what's going on, and I tried to explain things for an 8-year-old. Then he said, Daddy, are we going back to being slaves? This has been a tough week. And I just wanted to say that while I will try to focus in the next hour on science, there is a part of me that's very distracted and wanting to make sure that we all do everything that we can do to make sure that the lives of people like Ishmael and his son and their family and etc. are valued in the same way that other people's are. So with that, let's get to the science. Okay. So first, I wanted to start with the broad observation that the human brain displays an incredibly rich network architecture. This is a picture that has been constructed from diffusion spectrum imaging data, and it's a very high resolution scan that takes about an hour to complete for a healthy human subject. It maps out to 720 different diffusion directions, so it's super, super high resolution, much more so than the traditional diffusion tensor imaging data that's often used to understand white matter in the human brain. And this was a visualization that was constructed by James Bartolozzi, who was an undergraduate at the time combined between the visual arts and the sciences, and he now works at Pixar. We were lucky to have him for a few years making these beautiful visualizations. But I want to note that within that very rich network architecture, there are structural links that constrain the propagation of activity in a manner that is precisely dependent on the link topology. And I think that it's important in our effort to understand human brain and network architecture to realize that that architecture can provide clarity on the basic neurophysiology and also potentially inform neuromodulation therapies. Based on this structure, would we be better able to optimize and personalize neuromodulation therapies that account for individual differences and architectures inside? So in order to start addressing questions in this broad space, what I wanted to do was have a few preliminaries in terms of how we are going to synthesize the rich architecture and summarize the rich architecture that you see here. So what we will do over the next few minutes is to build a model of the brain as a network. In this case, regions of the brain will be a node inside of the network, and then structural links, so estimated white matter tracks, will be modeled as an edge inside of the network. And on the right-hand side, what you see is an adjacency matrix, and this is what we will use to visualize the structure of that network. So on the X and Y axes are the nodes in the brain, and so those are going to be the regions, gray matter regions of the brain. And then every element inside of this adjacency matrix tells you the strength of connectivity between node I and node J, or in other words, region I and region J. Now, after you've built a model like this, the very first question that may pop into your mind is, are there simple principles of network connectivity in neural systems? And we tried to tackle that question in a principled cross-species manner a few years ago, and this was work that was led by Richard Betzel, who was a postdoc in the lab at the time and has now started his own lab at Indiana University. And what you see here is that he collated brain networks, structural networks from several different species. The mouse on the left-hand side from the Allen Brain Institute, Drosophila, also sort of on the left-hand side, the macaque, and then 2 human connectomes from diffusion imaging. The low resolution one is where we have only a couple hundred regions, and the high resolution one is where we have close to 1,000 regions. So at the bottom of this slide, what you see is a plot for each of these different species and different connectomes, the weight of a structural connection and the distance of that structural connection. And what you can see is a very consistent negative relationship between these two variables. So that means that most of the connections inside of these connectomes are short, and that short connections are particularly strong. Now, what's a simple sort of generative model that might create that very consistent pattern over such diverse species and also over very different ways of measuring tracts or anatomical connections? Well, probably the simplest model that could account for that relationship is the principle of small worldness. And this idea was popularized by Duncan Watts and Steve Strogatz in their 1998 Nature paper, where they showed that you can take a regular network, which I'm showing you here on the left-hand side, and you can start to iteratively rewire edges in a random manner, and eventually you will get a random network. In between the very regular and the very random network is what's called a small world regime, where on average you have a few short paths that or a few long distance connections that can connect up one piece of the network to another. And that allows for global information transmission, while the short connections allow for local information processing. And so what's really nice about the small world network is that it has mostly short connections. The short connections are mostly strong, but there are a few long distance connections that allow for this global transmission of information. Now, what we decided to do in the paper was to ask the question is the small world network model enough to explain what we see in these several species? The answer is important. It's important to identify the answer, because if it's not enough, then we need to expand the model. It's certainly enough to explain that negative relationship between weight and distance, but is it enough to explain everything that we see inside of these connectomes? Now, to address this question, what we did is that we tested a prediction of the small world network model. That prediction is that the long distance edges are placed at random. Now, recall from the previous slide, the Watts-Strogatz model randomly rewires edges, and that's how you get these long distance connections. So if the brain was a small world network in the same way as the Watts and Strogatz small world network is developed, then you should see that the long distance connections are placed randomly. Now, for any neurobiologist or neuroanatomist in the virtual audience, you're probably going to say, of course they're not placed randomly, but let's hold on a second and at least statistically evaluate the prediction of the model. So in order to statistically evaluate the prediction of the model, what we do is that we calculate the similarity between node I's connectivity and node J's connectivity profile. If that similarity is greater than expected under the assumption that long distance connections are placed at random, then it would suggest that the small world model is not enough to account for what we see in real biology. So at the bottom of this slide, what I'm showing you is the actual data. Along the y-axis is the mean cosine similarity between node I's connectivity and node J's connectivity. And then along the x-axis is the percentile of long distance connections. So 5 means the 5% very longest connections, 10 means the 10% longest connections, 20 means the 20% longest connections, etc. On the left, we start with mouse, then we go to Drosophila, macaque, and then the 2 human. In color are the actual data from the true connectomes, and then in gray are the expectations of the null model that long distance connections have been placed at random. And what I want you to notice is that the colored bars on average are taller than the gray bars across all of the species. And so what that means is that we see much more similarity between node I's connectivity profile and node J's connectivity profile than expected under the assumption of long distance connections being placed at random, which means that the prediction of the small world model is not true. It's invalidated. So that suggests to us that we need to move beyond the small world model to incorporate more constraints or a richer parameter space for generative models of brain networks. Okay, so we can ask the question, what is precisely the rule for long distance link placement? If it's not a random distribution, what could it be instead? Now, some people in the literature over the last few years have suggested that a very simple rule for long distance placement would be to connect up one module of the brain with another module of the brain. So what I'm showing you here is that modular model. So on the left-hand side is a graph or a network that has modules being these clusters of nodes that are densely interconnected, and then the edges that go between the modules would be the long distance links. So on the right-hand side is an adjacency matrix representation of the same network that you see visualized on the left. And again, what you can see is that there's strong modular structure. That's the block diagonal organization. And then the long distance links are the bits that are colored in the off diagonal blocks. Now, if we were to evaluate this model in brain connectomes, we could do that by using community detection algorithms that are informed by a network prior. For example, modularity maximization would be one of those. But I want to ask the question of, is that enough? Is the presence of modularity enough to really explain what kinds of processes the brain might have to perform? And as we were thinking about it, we thought, well, I actually think that there are several distinct architectures, potential architectures, that would facilitate diverse information processing in a brain or other information processing system. So on the left-hand side is the modular one that we just talked about, but I want to complement that by two others. In the top, I have the bipartite structure, and in the bottom, I have a core periphery structure. Let's start at the top. So a bipartite structure is one where you have lots of connections between two sectors of the system, and that allows for very swift and direct information transmission. In the adjacency matrix, bipartite structure is often manifest in off-diagonal blocks that have high structural connectivity. So you see that most of the color in this matrix is off of the diagonal. And then in the very bottom, the core periphery structure is one that allows for the broadcast of information across the whole network and also the receipt of information back to some central portion. And so in the network visualization of that, you see a core, which is a densely intra-connected set of regions that also have strong connections to other portions of the network. In the adjacency matrix representation, what you see is one strong module along the diagonal and then a fall off of connectivity to the rest of the system. So that allows for broadcasting information to the rest of the system, but also receiving information from the rest of the system. And if you think about it, information processing systems in general might have to take advantage of all three of these different features. So what would happen if the brain actually displays all of those architectures? And in fact, what we find is that it does, and they're all superimposed on one another. So what we see in the brain is that it looks a lot more like this stylized example network, which has a core periphery structure. It also has a few modules, and then it also has some bipartite structure as well. So when you look at the adjacency matrix, what you see is that it's not just connectivity along the diagonal. There's really rich structures off the diagonal as well that allow for that information segregation, integration, transmission, receipt, and broadcast. Now, if we were to evaluate whether this was present in a brain, in a connectome from the mouse or Drosophila or the macaque or the human, what we could do is use what's called a weighted stochastic block model. And that is an algorithm that is tuned to detect blocks that have an even probability of connectivity inside of them, but doesn't assume that all of the strength of connectivity will lie along the diagonal the way that modularity or other community detection techniques tend to do. So the stochastic block model is a nice, much more sensitive and much more flexible algorithm that allows you to find a structure that's this complicated. So what we did is that we applied this stochastic block model to brain imaging data across all of the species, but I'm just going to focus on the mouse and the human for now. And to unpack precisely what we did, what I want to begin with is to start with the fact that this was structural connectome data from the Allen Brain Institute for the mouse. And we took the entire connectome, its structure, and we applied the weighted stochastic block model to identify where these white lines should be that would allow for distinguishing where blocks are. blocks that are modules, blocks that are bipartites, blocks that are part of a core periphery structure. And then what we did is that we asked, does that have biological validity? And to ask that question, we asked whether that architecture explains gene expression. So on the left-hand side is I'm showing you that where the white lines are placed as predicted by the weighted stochastic block model. And then on the right-hand side here, I'm coloring that matrix with gene co-expression. And what I think is particularly remarkable about this picture is that the gene expression was not used at all in defining where the lines should be. The lines are purely determined based on the structural connectome. However, what you see is that the white lines demarcate sectors of gene co-expression extremely beautifully. So what that suggests is that the structural connectivity inside of the mouse brain has an architecture that explains patterns of gene expression. And in the paper, if you're curious about the details, we go through a careful evaluation of whether this assessment of architecture is one that explains more about gene co-expression than simple modularity or other simpler models of the brain connectome. The answer is yes. But I just wanted to show you the visual so that you can keep that in your mind as I move to the human. So in the human case, we again take human structural connectivity and we apply the weighted stochastic block model to identify this sort of rich set of motifs for information processing. The white lines that I'm showing you here on the left-hand side are ones that were predicted by the stochastic block model. And then what I'm going to show you on the right is functional connectivity from the resting state of using human fMRI data. And again, what you can see is that the functional connectivity, first of all, was not used to determine where the white lines should be. Those were purely defined based on structure. But when you plot the functional connectivity on top of them, you see this really nice correspondence between where the white lines have been placed and the structure of the functional connectivity matrix. What that suggests is that the brain has a structural architecture that is quite rich. It doesn't just have diagonal blocks, it has off-diagonal structure as well. That explains patterns of functional connectivity in a really beautiful way. And again, in the paper, you can look at how we compared whether this was a better fit than other previous models for connectomes, and the answer is yes. But what I think is really important about this is that it suggests that we need to consider what kinds of structural motifs support information processing, and then use those to inform our analysis pipelines that we take to the brain. Okay, so that brings me to the second part of my talk, which is that now we've thought a little bit about the human brain's incredibly rich network architecture that is not perfectly explained by the small world model and is also not perfectly explained by the modular model, but really requires this richer set of motifs for information processing. We can then ask the question of how does that network architecture constrain the potential propagation of activity through this system? Now remember that all of these are white matter tracks along which activity can be propagated. So how does the network architecture constrain that propagation of activity? Now, before we begin this section, what I want to think about is what we currently have and what we really want to have, because there's always a discrepancy in science between what we have and what we want to have, and that directs our trajectories of inquiry. So what we have so far is a network of structural links that is empirically measured by neuroimaging. And then what we'd really like to have, what we seek, is a theory for how change in activity in one region affects activity in other regions. So what I'd like to do is to have a way to say, well, here is my brain, and then activity may be injected into a particular region of the brain. How does it flow through this set of structural links? And how does change in activity in one area alter the change in activity of another area, driving these alterations in regional amplitude with time? So to address that question, we began by formalizing the problem as one of network control. And this is drawn from work in systems engineering and the field of control theory, which asks the question of when you have a networked system and you have a change in activity of one piece of it, how does that affect the rest of the system? Now, what we did is that we started with a relatively simple model of dynamics in the brain, and that allows us to address questions of network control. I guess I will just mention here that many of the studies in using network tools to understand the brain have focused on statistics of connectivity. And so they've really focused on statistics of that adjacency matrix that we just talked about. And they don't necessarily combine that assessment with an assessment of the dynamics that occur on top of that adjacency matrix. And that's really what the formalism of network control allows us to do, is to take into account both the structure of the adjacency matrix, or the structure of the connectome, and also the dynamics that are happening on top of it. So these are the dynamics that we study. X of t+1 is the state of brain regions over time. So it's a vector. It's a 1 by n vector, where n is the number of brain regions, and t+1 is time t+1. We're going to say that that state of the brain is equal to the adjacency matrix A that we just talked about, times the state of the brain at time t, so x of t, and then plus the control. So B sub kappa is a matrix that has elements along the diagonal that tell you which region is being controlled, or which region has energy injected into it. And then U sub kappa is the control energy that's injected into those regions at a particular time t. So with this simple model, we can start to ask questions about what kind of control properties does a brain network have? And could we use those control properties to better understand how to optimize neurostimulation? So the first question that we asked is, which regions of the brain are most efficient or most difficult to control? And we decided to focus on two control strategies that have been proposed in the systems engineering literature, but hadn't yet been discussed in neuroscience, and I think are quite relevant to how we think about how the brain works. The first notion is the notion of average controllability, and that's the ability to steer the system into many easily reachable states. And the second idea is modal controllability, which is the ability to steer the system into a few difficult to reach states. Now, I probably won't go into the mathematics here very much unless there are questions about it at the end of the talk. But I just wanted to say that from that linear dynamics that I wrote down, you can write down what's called the T-steps controllability grammian, which is this W here. And then from there, you can extract these statistical predictors of whether a brain region is able to push the whole system to distant states on an energy landscape, and that would be a region that has high modal controllability, or if that region has the ability to push the system into lots of local, easily reachable states on the energy landscape, and that would be this notion of average controllability. So first we wanted to ask whether there are particular portions of the brain that are better average or modal controllers. And we started by thinking about the default mode and the sort of resting state of brain function. And we predicted that this resting state of brain function would be composed of areas that are able to push the brain into lots of easily reachable states on the energy landscape. And that is in fact what we found. So on the y-axis here is the controllability of these regions marked in blue. And then the two kinds of control that we study are the average controllability and the modal controllability. And as you can see, there's a strong preference for average controllability in those regions. What that suggests is that these areas that are active at rest have a particular white matter connectivity pattern that allows them to effectively drive the brain to nearby brain states. And you might consider that if you were to build an energetically efficient system, you might want the regions that are active at rest to be those that can enact these broad, most of the transitions that you would need to follow. Now, the next question is, well, what allows you to push the brain into a very different state? That's the modal controllability, at least in the systems engineering vocabulary. Which regions of the brain would we think would be able to do that? And it occurred to us that probably regions in frontal parietal cortex involved in cognitive control would be those that would have the capacity to push the system into these very distant states. So we tested that hypothesis, and here you see in green, the controllability of regions in the frontal parietal system. You can see a much higher amount of modal controllability there than average controllability. consistent with the hypothesis. So this suggests that regions that are implicated in cognitive control and executive function are those that are high in modal controllability and therefore structurally predisposed, just based on their white matter architecture, to push the brain into difficult to reach states. Now, I want to emphasize here that I'm not suggesting that cognitive control and executive function more generally are purely explainable by white matter tracks. We know that there are very important computations that are happening inside of the gray matter portions of these regions. But what I am saying is that their white matter structure is very helpful. Theoretically, it seems to be predictive that they would be able to enact this kind of control to push the brain into a very different state. And why do I think that that's relevant for executive function specifically? Well, because executive function and cognitive control, more precisely, are they're cognitive processes that are necessary for the control of human behavior, and it allows us to select and successfully monitor behaviors that facilitate the attainment of chosen goals. They are related to our ability to make difficult decisions, to inhibit inappropriate behavior, to have strong working memory performance, and many other features. So it's at least an initial suggestion that perhaps network control is a mechanism that affects cognition more broadly, but cognitive control and executive function specifically. But it's just the first study. And so what I want to do is to then suggest that, or state that we went on over the next few years to ask whether there was additional evidence in support of this idea that network control might be a mechanism for cognitive control. So the second study that we performed in this series is one where we studied the network architecture, so structural connectivity of youth between the ages of 8 and 22. And this is an age range over which executive function changes appreciably. And in fact, it's thought that adult levels of executive function are only reached around the age of 25. And what I'm showing you here is every single youth inside of this Philadelphia neurodevelopmental cohort group. There were 882 of them. And so every data point is one person. Their age is given in color, so from 8 years to about 22 years. And then the two axes are the modal controllability of that brain and the average controllability of that brain. First, what you can note is that the two of them track each other. So the brains that have higher modal controllability also have higher average controllability. So there does not appear to be a trade-off between these capacities. It actually appears that they happen together at the same time. They both increase with one another. Now, I also want you to note secondarily the color of the data points. So what you see is a strong clustering of the younger youth in the bottom left. They have lower modal controllability and lower average controllability. And you see a clustering of the older individuals, more in the bright yellow, in the top right quadrant. So they have high modal controllability and high average controllability. So this is consistent with the idea that network control capacities increase over this age range, just at the same developmental period that executive functions are changing as well. So that's another piece of the puzzle that is supportive of this hypothesis that network control could be a mechanism for cognitive control. Now, the third study was led by Eli Kornblath, and in this study what we did is that we considered these same youth and asked whether greater network control was associated with impulsive responses on a working memory task. And the answer is yes, so we drew a link between the actual behavioral performance on the task and the amount of network control for that particular individual. And that was an effect that held even when age was regressed out of the equation. So together, these three results suggest that our theory is a useful marker of how the brain enacts control to change network function. But I don't think we're done yet. And I think that there's one big open question in this part of the work that is about precision. So when I've been speaking about control, I've talked about the ability to push the system into lots of local, easily reachable states, or to push the system into a very distant, hard to reach state. But I've been completely agnostic as to what state the brain was in to begin with, or what state you're trying to go to. And I think if we really want to understand cognitive control and executive function, and eventually understand the use of neuromodulation to enact a particular state transition, then we have to have a model or a theory that's sensitive to this initial state of the brain and also the target state of the brain. So we began by building out our model a little bit more to account for these additional levels of complexity. So this was, again, some work led by Rick Betzel and also Shi Gu, who's also a professor now back in China. And what we did is that we took that model of network dynamics and we additionally defined a cost function for how a brain might move from one state of activity to another state of activity. That cost function penalizes the energy that's required for the transition. And so that's in the second term here on the right-hand side. And it also penalizes the distance of the current state of the brain from the target state. And that helps you to allow a smooth trajectory toward your goal. So if we minimize this cost function, it tells us where energy needs to be injected into the system in order to allow for a low energy and relatively straight trajectory from an initial state to a target state. And that allows us to ask much more complex questions about cognitive control. So here is the last sort of study in this part of the talk, where we wanted to probe the recruitment of brain regions in the executive system. So what we did is that we used this expanded model that allows for sensitivity to the initial state and to the target state. We started with an initial baseline state of no activity in the brain, and then we injected energy into the brain in simulation to allow a transition to the target state, which is frontal parietal activation. So activation of these regions that are important in executive function and in cognitive control. Then on the bottom here, what I'm showing is how much energy was actually needed for that transition, given each individual's white matter tractography. And what you see along the y-axis is the control energy, so that's how much energy was required for the transition. And along the x-axis is the age of the individual in years. And what you can see is that there's a negative relationship here, meaning that as youth age, they seem to require less energy to activate the frontal parietal system. And that's consistent with the notion that, if this frontal parietal system is easier to activate, then perhaps it's activated more regularly, and that explains the emergence of better executive function during this developmental period. Now, what we can also do is go one step further and say, well, is the amount of control energy related to individual differences in executive performance? So on the y-axis is the amount of control energy, and on the x-axis is the performance on a set of executive functioning tasks. And what you can see is that there's a negative relationship here, meaning that the people who score very well on these tasks that require executive function also require less energy to activate the frontal parietal cortex in our model. And people who perform worse on these executive function tasks are those that require more energy to activate the frontal parietal system in our theoretical model. We also find that the control energy can mediate the relationship, or statistically significantly mediates the relationship between age and executive function. Really, I think putting a finer point on the claim that network control is a potential mechanism for cognitive control. So putting these together, I think we can really say, and this paper was actually just published in eLife. It was led by Shai Zhu, who's a postdoc in the lab of Ted Satterthwaite here at Penn. And I think that these results are extremely exciting because they really allow us to move much closer to the actual activation processes that may be required to support executive functioning. Okay, that brings me then to the last portion of my talk that I wanted to share with you today, which is, now that we've seen something about the rich architecture of the human brain network and how that network architecture could constrain the propagation of activity in a good way, in a way that allows for complex processes like cognitive control. Now we can ask the question of whether those same principles of network control might be relevant for optimizing stimulation. And so I like to break down this question into two sub-questions. If we really want to understand the role of network architecture in optimizing stimulation, we sort of have to answer two things. On the left-hand side, are some brain networks more conducive to predictable changes in activity following stimulation? The answer is yes, but I'm going to skip that portion of the talk for today. If you want to see that work, it's published in Nature Physics in 2018. The first author is Jason Kim, who's a graduate student in the lab. The second question that we need to address is, are some brain regions better conduits leading to predictable states following stimulation? And do such regions differ across people? So let's tackle that question. To do that, we considered patients who have medically refractory epilepsy and who have therefore have electrocorticography sensors inside on the surface of the brain, and they also volunteered to be part of a grid stimulation study, meaning that they volunteered to have particular sensors on the brain stimulated, and then the activity following that stimulation processed and analyzed. And so what we did is that we studied in all 16 of these individuals who were willing to volunteer in the study as part of the DARPA RAM project. And what we did is that we calculated the modal controllability of the region that was being stimulated. So we have their white matter tractography. We also have their ECOG, and we know which region was actually being stimulated. So we can measure the modal controllability of the region that's being stimulated. And that's what's happening that's the variable along the x-axis here. And along the y-axis is the change in the memory encoding state probability. So what state of power across all of those sensors did the brain move to after stimulation? And is that state closer to their state of good memory encoding versus their state of bad memory encoding? And we obtained these estimates of memory encoding from a free recall task. Well, a whole set of memory tasks, actually, I should say. And I'll mention that a bit more on the next slide. But what I just want you to focus on now is that there seems to be a strong positive correlation between these two variables, suggesting that when you stimulate a region with high modal controllability, you push people closer to the good memory state. And when you stimulate regions of low modal controllability, you push people farther away from the good memory state. And so what that suggests is that these notions of controllability may be very helpful in explaining individual differences in response to stimulation, and particularly individual differences that are relevant for cognitive processes like memory. Now, the next question that we had was, well, can we really watch that change in brain state? So not just about average or modal controllability again, but broadening out to the broader model that allows us to say, here's a baseline state, here's a target state. How is stimulation helping enact that state transition? So again, we use ECOG data. What we have is data from the baseline before stimulation. We have a half a second of information pre-stimulation. We have the period of stimulation itself, and then we have post-stimulation, and then an interstim interval. Then from a free recall task, where this was work that was done by Mike Kahana's lab, they have extracted using a classifier whether or not the state of power across all electrodes is predictive of good memory encoding versus bad memory encoding. So we have, for every state that the brain goes into, we have whether it's close to good memory encoding or close to bad memory encoding. And then our question is, let's put these two pieces of information together along with the underlying white matter to say, well, now if we know what state the brain was in before stimulation and we know which region was stimulated, can we predict where the brain goes after stimulation? Now, to put that in a picture format, what we have is up here the pre-stim state that's up on the top left of the slide. We also have the diffusion imaging data that allows us to model how activity is being propagated through the network. And then we can ask two separate questions. We can ask about open loop control, which is just, if I stimulate this region, can I predict where the brain goes? And then we can ask about optimal control. Where should I stimulate in order to push the brain to a good memory state? And I'm going to sort of cut this portion short, but it is published just last year, 2019, in Cell Reports if you want to see all of the details of this study. But I wanted to highlight two pieces of evidence that suggest that this network control model is very useful in understanding the effects of stimulation here. So what we find is that when we enact this optimal control transition from the initial state that the brain was in before stimulation to a good memory state, we can predict how much energy was required. Now, on the y-axis here is that amount of energy, and then along the x-axis is the distance between the initial state of power across sensors and the target state of power across sensors. And there's a positive relationship here, which means that if you have to traverse a larger distance, so you have to go from an initial state to a very far state, then that transition requires much more energy than if you have to transition between two states that are nearby to one another. And that is intuitive, consistent with what we would imagine to be true, but it's nice to have it predicted by the model itself. And then on the right-hand side, what we did is to try to bring this a little bit closer to the cognitive processes that are at play. So along the y-axis, again, is the energy required for that transition. And along the x-axis is the change in the memory encoding probability. So what you see again is a positive relationship. Every line is a different subject, and then the black line is the group average. You see a positive relationship here, which means that the simulations that required more energy, the subjects that required more energy to enact this transition, were those who started in a state that was far from their good memory state. They were closer to their bad memory state. And then when we allowed this transition to happen, it required much more energy to get to the good memory state. Whereas people who were already close to the good memory state only required a little bit of energy to move all the way to the good energy state. And then we brought all of this together at the very end of the paper to say that we can actually predict with 93.2% accuracy the energy that's required for this state transition if we use three features: the distance, transition distance, so how far are you in your starting the state from where you need to go, the network topology of that particular person, and then the stimulation target, which was the region that was actually being stimulated. And so I think this data provides some initial evidence in favor of the idea that network control theory can be used to inform and possibly eventually optimize and personalize neurostimulation. I will certainly caution that this is a small data set. These data are hard to come by, but I think it's promising and certainly deserves more investigation in the future. So with that, I'd like to just sum up what we did over the last few minutes. We began with trying to understand how the human brain displays an incredibly rich network architecture. We talked about the fact that the small world model makes some predictions that are simply not true, and therefore needs to be expanded to account for more precise architectural motifs. We talked about the stochastic block model and how it allows us to detect three different kinds of motifs: modularity, a bipartite structure, and a core periphery structure, and that allowed us to better explain patterns of gene co-expression in the mouse and patterns of functional connectivity in the human. We then moved on to the second section where we talked about network architecture constraining the propagation of activity. And here what we asked is, well, let's not just focus on the architecture, but let's ask about the dynamics that flow on top of it. And we used a formalism from systems engineering called network control theory that allows us to really understand and make those questions more precise. We talked mostly about how that approach can allow us to think differently about cognitive control and executive functions. And then in the very end section, we talked about some initial evidence that suggests that these ideas from network control may be relevant for optimizing stimulation. And with that, I'd like to acknowledge a lot of the people who were important in this work, including the people in my team. A lot of this work was actually done by people who are now faculty. I tried to highlight a few of them as I was going through the talk. I also wanted to acknowledge several faculty collaborators. Ted Satterthwaite and Raquel Gurr gave us access to the Philadelphia Neurodevelopmental Cohort, which allowed us to test some of these developmental hypotheses. And then Fabio Pasqualetti, who's a control theorist at UC Riverside, side, and we've been friends since we were postdocs, and he's been a collaborator on all of the work that is using the network control model. And obviously, I'd like to acknowledge our funding as well. That's all I have. I would love to answer questions or exchange dialogue, etc. Thank you.
All right, great. Thank you very much. I am going to try once this time also. If you have a lot of noise going in the background, kids watching movies, if you want to turn it off, that's fine. I'm going to unmute everyone so we have an opportunity to give a applause, a round of applause for Danny's wonderful talk. I will mute everyone again, except for Danny. And we will get on to questions. Thank you very much. I know you only have, do you have 5 minutes, Danny, or?
I have 10 minutes, I think. Yeah.
Okay, great, thank you. So the question is, for those who can't see the chat box, have you tested this controllability association with executive function hypothesis using functional MRI data?
Using functional MRI data. So we have a connection between the yes, the beta weights. So what we did is that this is work by Braun, and it's on the archive preprint service. And what we did is that we took beta weights from a GLM, from fMRI data and to back working memory task, and then asked how much energy is required to go from activation of the 0 back to activation of 2 back, and then how much energy does it take to go from 2 back to 0 back. And we also we show that it takes a lot of energy to go to 2 back, takes less energy to go to 0 back. We also show that there is a what we call persistence energy. How much energy does it take to persist in a state? So can you persist in 0 back, or can you persist in 2 back, and how much energy would that require? What we find is that it takes much less energy to persist in 0 back, and it takes a lot more energy to persist in 2 back. We finally show that there's a difference in the amount of persistence energy that's shown in healthy individuals versus those who have schizophrenia and a known deficit in back-working memory performance. So that's work by Braun that might be relevant.
All right, we're going to move on to the next question. There's several of them, so I want to move in. Have an opportunity for you to answer as many as possible. Do you think that these energy needed for state transition would also hold true or same across various disorder or in healthy subjects?
Yeah, well, we definitely see that it's different in people who have schizophrenia. That's the Braun study. There's also a really nice study by Danielle Zoller and colleagues at EPFL that show that there's a change in the energy required for generic state transitions in people who have 22q deletion syndrome. And so that's one more piece of evidence that this is altered in clinical populations. Those are the only 2 studies that I know that use this method on patients that have psychiatric conditions.
Thank you. The next question is, the calculation for the Stiso et al. 2019 were done agnostically in regard to the specific brain regions, correct?
Yes, that's correct.
All right.
Well, well, let me just hedge that to say that we, when we're modeling it, we take the whole brain connectome as you see here, and then we inject energy into the precise brain region in which the stimulation was happening. So in that sense, it is we are taking into account the anatomy precisely.
Great, yeah. Thank you. Do you think the precision of diffusion imaging is enough for implementing network control in a particular regions or fiber in that sense? Say if it would be possible to implement the idea for deep brain stimulation.
I think, so certainly I think it has potential for something like deep brain stimulation. I think that it's always important in these discussions of potential to acknowledge that there are many different diffusion imaging sequences, and some of them are very high resolution, and those are the ones that you would want to use for the network control. And some of them are very low resolutions, and those you probably wouldn't want to use for network control. So I would just be careful in saying that, yes, I think there's a lot of potential, but I would emphasize the need to use the high resolution sequences.
Great. You mentioned the modal and average controllability of the default mode network and frontoparietal network. I was wondering what this is like for sensory networks like visual or somatomotor.
Yeah, they tend to not have as much control, which is interesting. But I think what might be going on there is that when we are estimating control, we are assuming that the brain is this isolated box, and it's clearly not. It's connected to the environment where energy is coming from. And so I actually imagine that we might see a richer picture if we add a node or several nodes that are the environment, that allow information to pass into visual cortex or into auditory cortex, and then also allow the brain to change the environment through motor cortex. So in other words, we could have 3 additional nodes inside of our network that account for the fact that the brain is not isolated. It is actually in contact with the environment. And I think that broader construction might be important in understanding the role of sensory motor cortex and visual cortex in control.
Great. 2 more questions. What do you think of a potential trade-off that arises like rigid thinking versus flexible thinking? I'm sorry, this is looking at your findings of increased network efficiency at older ages. What do you think of potential trade-off that arises like rigid thinking versus flexible thinking?
That's a really great question. We do have some work on functional brain flexibility and its relationship to cognitive flexibility. And then we also have work, and that's published. We also have work that's unpublished that relates brain flexibility to structural controllability. So I do think that there is a connection. So the way that your brain is wired to some degree constrains how flexible the dynamics can be, and how flexible the dynamics can be then constrain cognitive flexibility. Now, I'm drawing a very coarse picture here, but I think that there is a connection. The question is precisely how strong are those predictive relationships?
Great. Going back to the basics, one question was, how did you measure control energy again?
Yes.
Paper,
So in the very beginning, I had the first equation that I showed you was x at time t plus 1 is equal to the adjacency matrix times x of t plus the control, which was b times u. U was the control energy. So that's the control energy. What you do is that you run a simulation then that says, if I'm using these dynamics, how much energy do I need to inject, and into which regions and at which times, in order to allow for this state transition to happen. And that's the control, that's the energy that we're studying.
Great. One last question if you have time. Let's see, while this seems really relevant for higher resolution stimulation such as TMS, would it also be relevant for lower resolution stimulation such as transcranial electric stimulation, TDCS, TACS, and so on?
That's a great question, and I don't know the answer. I think all that the model requires is that there's a difference in the white matter depending on where you stimulate. And I think that's going to be generally true even over large sectors of the brain. But I think more work would need to be done to really understand how much the network control theory would buy you for something that is a very coarse stimulation. I don't know the answer.
Great. So I think we managed to squeeze everything in your hour, so you can go off to your next meeting. Thank you so much for joining today and last time as well. I know there's great interest. I heard from a lot of different places about wanting to hear your recording, so that was very nice to hear. And I'm sure it'll be the same this time as well. So thank you very much. And thank you everyone for joining. Hopefully you can join us in 2 weeks with Daphne from Columbia University. So thank you.
Thank you.